Hello, PsyQ community, and welcome to today's webinar, Viruses, Vaccines, and Inflammation, Neurological Perspectives. This will be a three-part series. This first part will focus on viruses with a special focus on human coronaviruses. Thank you for joining us today. Dr. Pattee is a clinical neurologist for Neurology Associates PC and is also Associate Professor and Clinical Instructor for the Department of Neurology and Co-Medical Director for the Muscular Dystrophy Association Clinic within the University of Nebraska Medical Center, UNMC. He also serves as the Clinical Instructor in Neurology for the Lincoln Family Practice Residency Program. Dr. Pattee has extensive experience conducting preclinical and clinical trial research, as well as hundreds of scientific publications, abstracts, and oral presentations.
Yes, thank you so much. And you set the stage for transitioning beautifully into our discussion on human coronaviruses, which is what we're going to dive into now. So as you see on this slide, there are quite a few different distinct circulating strains of coronaviruses, and these are not new. We're talking about them now because as of July 31st, 4.5 million people have been impacted and infected by this. So it's obviously something that we can't ignore. But these human coronaviruses have been around since the mid-1960s. And as you can see on this, Dr. Pattee or Gary talked about some of the highly pathogenic strains just a moment ago. So the SARS-CoV-1, the original SARS coronavirus, the MERS, and then the SARS-CoV-2, which is what we're dealing with now. But a lot of evidence existed before that for some low pathogenic viral strains. So typically when we're talking about low versus high pathogenicity, one of the key features for these low pathogen strains is that they really infect the upper respiratory tract and cause more mild to moderate type symptoms, just cold flu-like symptoms that are not often going to transfer into a more severe or complicated presentation, as opposed to the highly pathogenic strains, which is what we're so concerned about, that infect more of the lower respiratory tract. And this is where we can develop pneumonia and have some of those acute respiratory syndromes that Gary was just talking about. So as we build on this slide, I'd love for you to just share with our PsyQ community a little bit more about the structure of this coronavirus and some of the other key aspects. So as we transition and build to see the diagram and the additional points on this slide, you already mentioned the origination from bats. So the next few bullet points are dealing with some of that information, and it really plays a role in trying to develop treatments and trying to target different types of vaccines, which we'll be discussing later on. So Gary, if you could share some information as we get those additional bullet points for this slide.
Yeah, Rachel, as you mentioned, this is a very powerful slide, kind of busy, everybody. Let me walk through it as best as I can. Actually, we'll start at the top. Coronavirus. It was named a coronavirus because under electron microscopy, it had a crown-like shape, as you can see off to the right, a picture of the virus itself. Now, what we know about the coronaviruses as a class, there actually are three genera. There's alpha, beta, and gamma, but only the alpha and beta have been found to be infective in humans. And as Rachel mentioned, and the slide points out, there are 7 distinct circulating strains. Now, these are very large single-stranded RNA viruses that we're dealing with, and the low and high pathogenic varieties are mentioned there. 2 of the low pathogenic have been known to invade the nervous system, which we'll talk about a little later on as well. But I think it has to do with the size of the virus. The smaller the virus can become, it can get deeper into our respiratory tract and potentially constitute a more pathogenic variety. The bullet points refer to bats and human coronaviruses. You know, with the SARS-1, the palm civet was the intermediary host between the bat to the human. And with the MERS, it was the dromedary camel. That was from the Arabian Peninsula to the UK. So it's interesting how, from this zoonosis perspective, as we interact more with animals as humans, there's the potential for more transmission of viruses in different organisms. You can read the last 2 bullet points that there's genetic similarity between the SARS-CoV-1 and CoV-2. Interestingly, one of the features that separates these 2 viral strains happens to lie on the receptor binding domain, and that certain actually treatments for the SARS-CoV-1 haven't been effective for the CoV-2 because of this particular reactive or receptor binding domain difference. And the mortality rate we have as the last bullet point, and that's our current data, up to 2 to 4%. Kind of jumping over to the right side, one thing we'll point out and we'll speak a lot about is the S protein on these coronaviruses, referring to as the spike protein. This is basically the lodging device that attaches to the ACE2 receptor on the epithelial or endothelial cell within the human. And it mediates entry into these into the organism. It requires the receptor binding domain as well. So under the S protein, 2 bullets, second bullet, critical target for neutralizing antibodies. And that's key. A lot of the neutralizing antibodies are thought to potentially affect the receptor binding domain of this entire mechanism of the S protein, spike protein interaction with the ACE2 receptor. And we'll review this a bit more. And you can see other protein components on this coronavirus that aren't quite as pathogenic, we may not know as much about. But the spike protein is really the focus of what we'll be discussing and what a lot of the vaccines may be focusing on as well.
Absolutely. I think that's the critical point for everybody to appreciate is some of the neutralizing antibodies that have worked for the original SARS have been tried for this novel coronavirus. And because of that spike protein and not being able to bind to that, they're not efficacious for fighting this novel coronavirus fight that we're doing now. And I'm sure you and Jackie will talk more about that with the vaccine discussion. So let's shift gears a little bit and talk about the infiltration that this novel coronavirus takes to get into the human host and some of the caveats there, where we're going to introduce another key player that people have probably heard a lot about in the media and would like to make heads or tails of, and that's the ACE2 receptor.
Yeah, and I think, Rachel, to look, this is a very important slide, and I'd like to start again on the right side. Let's just read through the bullet points. The coronaviruses enter the body via the respiratory tract, and that's very important just to emphasize. Thereafter, they attach to these ACE2 receptors to infect the human host. Second point, these receptors are cardiocerebrovascular protection factor existing in a variety of organs. And we'll talk about where these ACE2 receptors are localized. They can regulate blood pressure and anti-atherosclerotic mechanisms. Now, coronaviruses may cause abnormally elevated blood pressure and increase the risk of cerebral hemorrhage via binding to these receptors. So there are some vascular risks that we are aware of. There's something that we've seen as the COVID toes, that there's a lot out in the literature about bright red toes or discolored toes, likely as a consequence of these ACE2 interaction with the vascular regulatory mechanisms. And the last point is the spike protein, once again, can interact with these ACE2 expressed in the capillary endothelium, attacking the blood-brain barrier to enter the CNS. And there's also interaction with the epithelium. So jumping to the left side of the slide, the cartoon shows the coronaviral infection entering into the human host, and the blue dotted line takes it down into the respiratory tract. There's another upper blue dotted line that tracks along the olfactory bulb starts the olfactory nerve, through the cribriform plate, the olfactory bulb, and then infecting the brain. And just focusing on the bottom portion first, you see it says coronavirus. It attaches through the spike protein to the ACE2 receptor, entry into the cell. It's oftentimes taken up by the endosome, which is a part of the cellular debris mechanism. And within the endosome, there can be a series of reactions, the uncoating of the RNA, replication from the endoplasmic reticulum and the Golgi. We get protein production, reassembly, and that actually can lead to release of a newly formed coronavirus into the system. But you can also see a little bit of a red line going from the lungs to the brain. That constitutes the bloodstream. So the virus through the lungs can enter the bloodstream and go anywhere. And in this particular instance, it can travel to the brain by that mechanism. There's also the vagus nerve on this slide. As most of you recognize, the vagus nerve is the autonomic nervous system, cranial nerve 10. It has sensory fibers, however, within the trachea, the larynx, the lung tissue. And so virus particles can be taken up through a retrograde transport mechanism and thereby also delivered to the brain. And then the top cartoon just shows through the blood-brain barrier, or the CSF on the top, that once inside, the coronavirus can have an effect on the neural tissue, whether it's a neuron, oligodendrocyte, astrocyte, the microglia. You see a host of inflammatory reactions that take place. And usually it's the more severe infections that get into the CNS, which we'll refer to a little later on. But this is a little bit of an overview of how the host is infiltrated by this virus.
Thank you so much, Gary. Yes, and for those of you who have been following some of the literature and at least some of the areas of interest that are starting to be burgeoning throughout our scientific field is related to the microbiome. And we know that ACE2 receptors are also expressed on the gut epithelial cells. And we see about 40% of COVID patients are showing significant GI symptoms as well. So just to kind of piggyback on what Gary was saying, the gut-brain axis may be another mechanism by which the coronavirus makes its way into the central nervous system.
One other clinical point. There's been a lot of discussion regarding the use of ACE receptor inhibitors, ACE inhibitors, as well as angiotensin receptor blockers, both medicines for blood pressure. There's been some concern that would they, because both of these agents upregulate the ACE2 receptors, would they potentially make it more likely for somebody exposed to COVID to become sicker potentially, or even more severe cases? And the studies so far have not shown that. We have not seen that individuals on ACE inhibitors or ARBs for blood pressure have a higher risk or mortality if infected. So that's a question that has been asked quite a bit, but to date, that has not been considered a risk factor.
So let's wrap it up. What are your closing thoughts on all of this, Gary? Where are we? Where are we going to be? Can you look into your magic ball and let us know how this is all going to turn out?
Well, I wished I had all those answers, Jackie, but let's just spend a little time going through. We covered a lot of ground here today, but let's just highlight it bullet point at a time. So we talked about 7 distinct circulating strains of coronavirus, 3 being highly pathogenic, the COVID-19 being one of those. Bullet point 2, coronaviruses enter the human host via the respiratory tract. And we can stop there for a second and just focus on transmission. Respiratory droplets from face-to-face exposure. And how does that occur? Well, coughing, talking, sneezing, most common mode of transmission. And it's important to point that out because I'm a big advocate for the masks, even though I kind of detest the masks myself because I'm always coughing or sneezing. I saw a cute meme that says, if you don't like masks, just imagine how you'll feel with a ventilator. And I thought that was a good description. Yeah. From exposure to symptom onset is oftentimes 5 days with this virus. So you could go several days without knowing it, but still you could be infective. And then there's statistics saying that by 97% of people develop symptoms within 11.5 days. So it may be anywhere from 5 days to a week later before you get the symptoms, all of which you could be infective, or another individual next to you could be as well. So I just don't want to underemphasize the aspect of the respiratory tract as in the human host mechanisms with this virus.